Hi, Max. This is Merritt. I just wanted to go through what the poster would be like. So let's go to your abstract real quick and take a peek at that. So if we're looking at this, whoops, wrong thing. Here we go. If we're looking at your abstract, basically what we're driving at is this title saying phosphomimetic mutation of NATO3, a floor plate BHLH gene drives expression of neuroprotective genes, FOXA2, GRAIL1, and NER1. And this neuroprotection angle is the one that we're going to try to present and bring sort of to the fore in this in this work here. You can hear Patrick in the background. So let's kind of hit it and get into what that looks like. You'll change the abstract, you'll change that point. So here, our angle is kind of going through our story a little bit about how we came across this. What I would probably do is at the intro, basically say that introduce this where NATO3 is being expressed, which is in the floor plate, in the developing midbrain and spinal cord. And so it's expressed sort of in this domain. That's where the midbrain is, and in the embryo. And it's right along that ventral midline. So that's the thing that we're going to want to, so that's where it arises, and that's where dopamine neurons arise. And so because NATO3 is expressed in those cells, we were curious to see whether it was important and sufficient to drive genes that are important in dopamine neurogenesis. And interestingly, a lot of genes that are important for dopamine neurogenesis and development are also important for neuroprotection. So we wanted to, capture some of that story, or at least some of those, and see whether NATO3 is actually sufficient to be able to drive those that gene and that expression. So the story that I'm going to tell you in this poster basically is saying, the first part of it is going to say that we've found that the wild-type NATO3 can help drive some of the genes that are important for dopamine neurogenesis, including NER1, but only in certain regions. And that drove us to ask the question whether we could expand this capacity of NATO3 to induce dopamine neuron gene expression much more broadly, like in the telencephalon and also in the spinal cord. And so we modified this gene mimicking phosphorylation events that we speculated may be taking place within the midbrain itself. So that's what, and so we did that. We've developed the tool, and then the takeaway basically is that this tool seems to be able to drive dopamine neurogenesis and those dopamine neuron markers broadly. And right now, my work is picking up on that and trying to characterize what kinases are responsible for causing this kind of event to happen in vivo, and how can we manipulate it. So let me kind of just go through this a little bit in more detail with you, if you have time. So you're like, oh yeah, sure, let's walk through the data. Great. So in that point, you've given them a little bit of a snippet. They have an idea of what you're up to. They can walk on if they want, or they can get more engaged. So, all right, fine. They're going to talk a little more. Then you say, okay, well, essentially what we're looking at then is a couple of, this summarizes that NATO3 is a VHLH. It's expressed in that floor plate, like I told you, and that we're looking at how it's being, and that its action is in the midbrain. So the first thing that we've been testing, and what we did, is that we did a series of studies, and we're going to focus on this part of the methods right here first, and pass this for a second. And we're gonna, and what this basically is doing is, it's our methodology was basically trying to test our question of, is NATO3 sufficient to drive dopamine neuron marker or dopamine neuron gene expression? And so we injected the NATO3 plasmid within the lumen of the developing neural tube of the embryo. And that's a bicistronic vector, so it expresses EGFP along with our gene of interest, and so we can visualize where it's actually being expressed within the developing embryo. So here's the eye, here's the forebrain, and this is the midbrain right here after we allowed it to incubate for 66 to 72 hours. And then we section the embryos. To see what was happening and what kind of changes were actually occurring. And we can trace to see with the green cells, have they actually changed their behavior relative to the control side. And this control is actually an internal control. It's nice because these are unelectroporated cells over here. Additionally, we can do things like quantitative PCR and be able to characterize it that way. So let's take a look at what we saw. With the wild-type NATO3, what we basically found was that if we, on the treated side, the part that was getting electroporated on the chick embryo after we allowed it to develop, we saw greater numbers of Neur1 positive cells in here. And you can see that this is, red represents Neur1. And so we can see that expression there, and much more greater numbers here on the electroporated side. And what we can also see is that if we do look at a close-up, that basically everything that's green is also red in this case. So it looks like the greater numbers of these dopamine neurons that have arisen in the treated side are due to the effect of the electroporation. So this is that wild-type NATO3 that's got us really interested in it, and it was interesting because some regions of the caudal portion of the ventral midbrain, like where we have this section right here, we could really see a lot of the effects of the wild-type NATO3. But when we section more rostrally up here, we just didn't see that kind of effect here, and so it wasn't quite as robust. So these data then suggested that we were looking at how. So these data suggested that wild-type NATO3 acted in a really specific region, but we wanted to kind of open it up a little bit and see if there was anything that we could do. So we tried a lot of different types of tests that aren't shown here, where we co-transfected multiple different factors along with it. But one of the things that we did is that we mutated the phosphomimetic NATO3. So I'm going to go back up to here and show you that the sequence alignment for NATO3 here shows a conserved threonine and a serine residue here. These are ones that are important for the development, or these are ones that are important in other basic helix to helix proteins such as ASCL1 and OLIG2. So in those particular proteins, if you mutate them, you can really change the function of the developing of how ASCL1 and OLIG2 work. So we wanted to see if we mutated it, how it would work in the embryos. So what these data show is basically how overexpression of our phosphomimetic NATO3, where we've mutated the serine and replaced it with an aspartate. What we're finding is that there's a greater number of Neur1 positive cells that aren't seen in other regions. So it's much more rostral, like where we have over in this domain over here, we start to see this Neur1 positive cells appearing there. So that's one of the phosphomimetic mutants that we were working with that showed that. Here's another one in which we actually saw this the same similar types of changes happening with other markers such as sonic hedgehog and LMX1B. And in addition, if we look down here, in the treated side of phosphomimetic NATO3, we have engrailed 1 being shown at really high levels on there. So on this side, on the transfected side versus the untreated side over here. So here we have PM-NATO3 really highly expressed, for example, and we can see that there's a lot of overlap between these two conditions. So these are all sections of tissue from the midbrain looking at how these effects are happening. They're really robust and strong, estimating around 90% or so of co-expression of these different markers. So that's pretty powerful in terms of its effects. What we wanted to do then was to look to see what how it worked in other areas of the brain as well. So we have it here in the induction of FOXA2. So this should say FOXA2, and this should say FOXA2. FOXA2. And what we're seeing basically is that there's a really robust induction of FOXA2 throughout the nervous system here. So that's pretty interesting as well. So this is pretty cool because this is a cross section of the spinal cord, and what we've done is that we've green here represents the overexpression of the NATO3 wild type NATO3. This is demonstrating the overexpression of the phosphomimetic NATO3. You can really see how broad and robust that expression is. Finally, then, if we look at what happens with phosphomimetic NATO3 and that rostral portion of the midbrain, we can see how FOXA2 is really highly induced there. So what's interesting in terms of, how generalizable is this type of phenomena? Is it only something that happens weirdly only in the chick? And so when we looked at this SN4741 cell line, which is one that is a midbrain cell line, what we found was that there was a really large induction of LMX1A and NEUR1. So these are two different genes that are associated with the expression of with neuroprotection and with neural development. So my current work is trying to put a bigger context around this. So we found something that looks really interesting. We made a speculation that this modified NATO3 gene could be promoting dopamine neuron marker or dopamine neuron gene expression, including these neuroprotective genes like engrailed1 and FOXA2. But now what we're looking at is that we want to be able to say and understand a little bit more about what's happening with the cells and whether what kinases are responsible for dictating this effect, and what are the sites on NATO3 that are actually affected. So I'm currently working on developing, and we're working on overexpressing that catalytic subunit of the protein kinase A. And what we found basically is that if we have PKA and NATO3, so this kinase plus NATO3, we see a really big induction in the expression of engrailed1 and in some other markers. It doesn't follow quite, exactly to form to what we saw. We don't see a big induction of FOXA2, for example. So there could be some differences that are happening with our cell line versus what we have when we're looking in vivo. But what this really does indicate is that the phosphorylation conditions that we see around NATO3 could really be important for helping to dictate it. So this is preliminary data. We're repeating it now. So we're just in the midst of that process now. But I wanted to. But it's what we're excited about, and will, I think, provide a nice context for us to be able to appreciate how this gene actually works. Okay, yeah, so let me see here. Let me go back to it. Now, where was this section from here? Where's this caudal ventral midbrain? Oh, it was right back in this region here. So we just crossed through the sections, and we did, what I mean by that is that we sectioned this tissue, and so that we're looking at these transverse sections through the midbrain here. And so, oh, okay, all right. And so you think this is different? And I'm like, I don't know. I mean, these are kind of less here than you have over here. Is that meaningful? Could be. When we looked at this overall, we really didn't see a big difference between the unelectroporated side and the electroporated side. And in particular, that key point is that we're just not seeing that induction of NATO3 for sure. Okay, well, all right. Let's see, what else do I really think about this? So, tell me about neuroprotection. I don't understand it at all. Well, there are a lot of aspects of neuroprotection that are still being worked out. But one thing that's been really interesting is that if we look at what happens with engrailed here and this induction of engrailed, what's so exciting is to think that this phosphomimetic NATO3 may be sufficient to drive a neuroprotective gene. Engrailed has been purified as a protein and injected into the striatum, or the developing, or rather the substantia nigra of these. Maybe it's a striatum, I have to check. But into the brains of animals that have been haplosufficient for engrailed, and that's been able to be to help correct for some of the challenges that those animals have. And so it's been shown to be a nice neuroprotective factor. So what's really kind of cool about this is that NATO3 is actually able to help promote the protection of, promote and drive the expression of a lot of these different genes that are important in neuroprotection. Well, okay, I see what you're saying here. So, and now you're really hopeful about this thing here. Yeah, we're looking for that. What we've done is we looked at some time points we're not seeing that come up, and other time points we are. And so we really want to make sure it's reproducible. But we're excited about this. It fits the story really well, and I thought it would be something that I could include in the just as a talking point in the conversation that we're having. Okay. So what do you think the next step is? Well, the next step I think would be, in this context, is to see, well, what's happening? Can we understand a little bit more about how NATO3 is actually driving this? Is NATO3 expressed in adult neurons? Interestingly, no, it's not. So it's not something necessarily that we can just activate in adult neurons. We'd have to use this in some way as a tool, introducing it into the adult neurons of sick patients, or something like that, or perhaps introducing the protein into the neurons of sick patients to try to help with and correct for some of the challenges that they're facing. Oh, okay. So what are you doing to test that? Well, other people in the lab are working on and collaborating with the Brendan lab to be able to characterize the effects of this PM-NATO3 in cell culture using human dopamine neurons called LUMIS cells. And so they're testing that to see whether just this uptick in overexpression of these different neuroprotective markers would be sufficient to help protect them against environmental toxins like MPP plus or against alpha-synuclein as some examples. Oh, okay. That's interesting. How is that going? Well, it's all, it's just that work's just getting underway. So we'll see. We're excited to see what the results are. Okay, well, that's really interesting. So what did you do? So this poster is reflecting a lot of our work that we've been doing in the lab and provides a lot of context for it. So my work's been looking at these a lot of the cell culture, and in particular working on this aspect here. Since I'm an undergraduate in the laboratory, my work, the scope of my work is really pretty focused, but we work together as a team to be able to help solve these kinds of problems. Oh, okay. Well, that's really interesting. That's great. Well, you should come to my medical school. Oh, okay, great. That sounds good. Where are you at? I'm at Harvard. No, that's great. I've heard of it. Okay. So you get the idea. I hope that helps.